people are gathering their food. We cannot start the seminar without food and coffee. I will go ahead and introduce Bryan. So please settle in as soon as you guys can out there. Um, it's my great honor and pleasure to introduce you to Bryan Gitschlag, who's visiting us from Cold Spring Harbor. He's currently a postdoc there and he's starting a new position at University of South Carolina, the other USC on the other side of the coast. And so I first met Bryan when I gave a seminar at Vanderbilt a number of years ago, which was pre-COVID. And you'll hear about the work that he was doing there. He comes from an intellectual lineage that includes Hamid Malik, which you'll see some of the interested selfish elements coming through there. And as Bryan contacted us since he's starting at his lab, he wants to use some of the approaches and technologies that my former graduate student, Zach Stevenson, developed while he was a PhD student. And Bryan's not going to talk about that, but I thought it'd be great for him just to come out so that we can talk one-on-one, and he gave a presentation in my lab about kind of the future directions. So it was good practice for reusing those both the current job talk and the future directions. And so we'll have the lunch with the graduate students right after this talk. And this is a great opportunity to talk to someone who just on the job market in probably the worst possible year. And so if you want to chat with Bryan about what that felt like and how those approaches work, then that could be a good opportunity in addition to the science stuff. So as I said, Bryan's at Cold Spring Harbor, where he's in Dave McClandish's lab, and he did his PhD at Vanderbilt. But he's actually from the Pacific Northwest originally, from Washington, and then he actually went to George Fox University outside of Portland for his undergrad, and he did undergraduate research at OHSU. So he's definitely familiar with Oregon and the environment that we've got here. And I think with that, because we've already cut into his time with our snacks, I'll just turn it right over to you, Bryan.
Okay. Is this a good volume level and too loud, not loud enough? I'm getting a thumbs up in the back. Okay, cool. Well, thank you so much. It's a little bit of I think maybe kind of scandalous that I'm here because I come from a family of beavers, but go Ducks. So thank you everyone for being here. I'm really excited to kind of share about some of the science journey that I've been on. And I'd like to start by basically orienting everyone to kind of like the big picture of what my interests are. And that has to do with the fact that living systems, especially complex systems, show a combined effect of multiple different evolutionary forces. And one good way that I think to kind of illustrate that uses one of my favorite types of organism, parrots. And I have a number of birds. I love birds. And I think what's really interesting about them is that they have evolved under many parrot species, at least, have evolved under selection simultaneously for attraction and for camouflage. And I think what's really interesting about this example is that it kind of illustrates that that it's not just a matter of one selection pressure winning out over the other, but rather that different evolutionary forces combine to create something new that we observe in nature. So for part one of my talk, I'm going to be discussing kind of similar concepts, but in the realm of evolutionary molecular biology, as it pertains to evolutionary conflicts, specifically in the case, as Patrick mentioned, of selfish genetic elements, and that relates to this phenomenon of multi-level selection. And to kind of illustrate what I mean by that, let's suppose that we have two genotypes that occupy or inhabit the same organism, where red has a replication advantage over blue. However, the selection pressure can flip the other way if you're looking at a different level of biological organization. So for example, if an individual that has more copies of blue has a higher replicative advantage, then which genotype has an advantage over the other depends on which level of biological organization that you're looking at. And so in the first part of my talk, I'll cover my work on how these different evolutionary factors like these combine to explain the genotypes that we observe, particularly in the case of selfish genetic elements within animal mitochondria. And of course, genotypes are not just the products of selection, right? But an interplay between mutation and selection. And different mutations can occur at drastically different rates, as represented by the variation in the thickness of these arrows. And these biases in mutation can vary between species. And as I'll cover in the second part of my talk, what I found was that during my postdoc work, that variation in, or species differences rather, in the genetics of adaptation reflect differences in their underlying mutational biases. And then finally, toward the end of my talk, I'll be talking a bit about my ongoing and future work, which combines elements of parts 1 and 2 of this presentation by integrating information about the rates and the effects of different mutations into more comprehensive roadmaps of sequence evolution. And to do this, I model evolution on fitness landscapes, which map fitness values onto all genotypes within a defined sequence space. And these fitness values can be obtained from empirical data, especially with the development in recent years of these large-scale, high-throughput mutagenesis assays, which can measure phenotypes for up to thousands or even millions of individual sequences. And very briefly, the way this works is that a sequence library is introduced typically to a cell line, followed by selection. And then we have a procedure for inferring the structure of a fitness landscape from all of those measurements of fitness. And in my postdoc lab, we use computational methods for inferring the basic structure, the geometry of the fitness landscape, and that can tell us some very useful, intuitive biological insights about how sequence evolution works for these sequences that are defined by the sequence library. And existing approaches, as illustrated here, existing approaches for inferring empirical fitness landscapes typically involve single-cell growth assays. And while this is highly informative and useful in telling us the molecular details of genotypes and molecular level phenotypes, these experiments don't capture the complexity of multicellular organisms. So one of my big goals moving forward is to take this general workflow and adapt it to an animal model. So here's the single cell illustration. And then my goal is to adapt this to an animal model, which is now possible with recent developments in high throughput approaches for precision genome editing, that is to say, developed here at U of O in the Patrick Phillips group. Okay, so backing up, I'd like to start with an overview of my past work on selfish genetic elements in mitochondria. And this represents an area of continuing interest between my graduate and my postdoc work, all of which is out now in the literature. So feel free to take pictures if you're really interested in this part of the talk. Okay, so what are selfish genetic elements in the first place? And to help answer that, I'll go ahead and say that they represent molecular level examples of a broader phenomenon within biology, where you have complex biological systems which are predicated on cooperative interactions, but are also vulnerable to those cooperative interactions being parasitized by what we could call selfish or cheater entities, which manifest at different levels of scale or different levels of biological organization. So in addition to selfish genetic elements, we also see similar examples at the cellular level, and as in the case of cancer and dictyostelium slime molds, you may be familiar with that example, and at the level of social animals. And so one of my big picture interests has to do with this nature of cooperation and conflict resolution as it plays out in evolutionary time. And in order to really kind of understand the evolutionary theory behind cooperation and conflict dynamics, it's important to have an experimentally tractable example. And so that's kind of what sort of hones my interest on mitochondrial genetics. And so on that note, as you may know, mitochondria are descended from a bacterial ancestor that became part of the eukaryotic cell through an endosymbiosis event, resulting in what has been described as the most successful mutualism in the history of life on Earth. However, due to their bacterial ancestry, mitochondria continue to contain their own genomes. And the mitochondrial genome, at least in mammals, contains 37 genes, which includes 13 proteins and 24 RNAs. And all of these are essential. They play essential roles in the electron transport chain. So mutations in mitochondrial DNA are implicated in a variety of diseases, as well as just the general process of aging. Now, other than being a small circular genome, mtDNA actually differs from the nuclear genome in a couple of other important ways. So the mitochondrial genome, for example, is a high copy number genome, so that a typical animal cell contains multiple individual mtDNA molecules. And what this means is that when a new mutation arises, it gives rise to what we would call a heteroplasmic state, where you have mutant and wild type copies of mtDNA that coexist within the same cell. And initially, that mutation is present at low frequency. And so even if it disrupts an essential gene, the organism might not feel those effects. However, another difference between between mtDNA and nuclear DNA is that the replication of new copies of mitochondrial DNA is not tightly coupled to the cell cycle the way the nucleus is. And this can enable selection, and it can enable the mutant mtDNA to vary in frequency over time within individuals, as well as between cells within an individual. And if the mutant mtDNA levels rise high enough, then they can cross a critical threshold, at which point a previously negligible mutation can become pathogenic. And so a big question that really drives my interest is, how does selfish mtDNA propagate? And in order for that, we need not only an experimentally tractable system of a cooperation and conflict dynamic like we see in mitochondria, but we need an experimentally tractable model that we can actually bring into the lab and study. And Caenorhabditis elegans is really an excellent model system for this type of research for a number of reasons. One is that the female germline is this vast syncytium of shared cytoplasm inhabited by multiple, up to thousands of individual nuclei. And so you have this really large contiguous cytoplasmic pool of mitochondria within the worm. And that actually represents the vast majority of the mitochondria within the worm. Over 90% of the mtDNA in the adult hermaphroditic nematode is confined to the female germline, which is precisely where mtDNA genotypes are competing for transmission to the next generation. So most of the population dynamics that we detect for mitochondrial mutations reflects the biology of the female germline. And in addition, we have the benefit of working with a really well-characterized example of a selfish mitochondrial genome, UADF5, which is represented by, it's a deletion represented by the red bar here, and that disrupts 3 out of the 5 electron transport chain complexes. And as you might expect, that leads to deleterious organismal phenotypes, as characterized by actually a drop in respiration rate, oxygen consumption. And that actually coincides with an increase in the overall amount of mitochondria. So the worms have more mitochondria, but those mitochondria are functioning suboptimally. And that results in a lower rate of fertility, a lower egg-laying rate. And in spite of that, this mutant mtDNA variant is able to persist at high frequency, representing upwards of 60% of the total mtDNA within a heteroplasmic population. And it's able to persist at this high frequency by out-replicating or out-competing the wild-type mtDNA. And so from over the course of development, we see a rise in the average frequency of the mutant genome. So how does this work? And in order to really address that question, I started to try to understand, what are the sort of ways that this mutation might be impacting the physiology of the organism that gives it an advantage. One possibility is that this mutation may be somehow evading organismal selection through something that it's doing to the physiology of the organism. And so on that note, one of the first things I looked at was the activation of stress responses that might be present within the organism. And indeed, we found transcriptional upregulation of stress responses that are indicative of this pathway, the mitochondrial unfolded protein response. And that gave us some follow-up hypotheses to test. And so, for example, what happens if you inactivate the unfolded protein response? And what happens is that the mutant mtDNA levels actually come down now. And alternatively, what happens if you take animals that have low initial levels of the mutant mitochondrial genome and you constitutively turn on the mitochondrial unfolded protein response and you see the mutant mtDNA levels rise again? So back to that question of how does selfish mtDNA persist despite its deleterious effects? There's more, it's a lot more complicated version of the story, and I'd be happy to talk more about it later one-on-one. But the general picture looks something like this. You have a heteroplasmic state where under normal conditions, the symbiotic relationship between the mitochondria and the nucleus looks like this, where you have the mitochondrial genome contributes to energy production, right? And if you have a mutation that disrupts mitochondrial function, that might contribute some form of metabolic stress or mitochondrial stress that the nucleus then is transcriptionally responding to. And one of the ways that the cell responds to mitochondrial stress is to make more mitochondria. And that can actually lead to this vicious cycle where you have an increasing amount of mitochondrial stress due to increasing levels of the mutant genome, driving a vicious cycle. And okay, so based on this was kind of closed out my first project in grad school. This led to sort of the overall conclusion that we have this interesting situation where the mutant mtDNA is able to outcompete the wild type within the worm. However, worms that carry lower frequency of the mutant mtDNA seem to be healthier. And so presumably at the level of organismal selection, then selection should favor the wild type mutant mtDNA. And this became really important to me because one of the big goals that I had in grad school was to try to uncover conditions that would allow me to, or try to discover conditions rather, that would allow me to uncover animals or recover animals that carry lower levels of the mutant mitochondrial genome. And so one example, I don't really have time to get into, but we found that if you inactivate the insulin signaling receptor, DAF2, in the worm, then you recover a population of worms that carry on average lower levels of the mutant mitochondrial genome. And the reason this multi-level selection paradigm became important is because we might want to know, for reasons, of trying to translate this into a mammalian system, we might need to know where is that signal coming from. Is the loss of insulin signaling just simply making the animals that carry high levels of mutant mtDNA less viable, and therefore we're just inducing stronger organismal selection. Alternatively, the loss of insulin signaling could do something interesting to the physiology of the organism that deprives the selfish mitochondrial genome of its replication advantage. So in order to really follow up on those questions, it actually became important to develop a quantitative description of how these different evolutionary forces combine under any given condition. And so I actually ended up going down this road on this project of designing experiments to measure the strength of selection at different levels of biological organization in isolation. And so starting with intra-organismal selection, this is the level where the mutant mitochondrial genome has an advantage over the wild type. And the way I came up with to try to measure that selection in a way that's isolated from the effects of organismal selection was to simply compare the mutant mtDNA frequency among a pool of siblings, sibling progeny, against their own individual parent. And I did this in a number of different isolated parent progeny lineages in order to minimize the confounding effect of selection between different parent progeny lineages. And if you do this experiment, then we see a really nice, clean uptick in the mutant mtDNA frequency on average from any given parent to its own progeny. Now, measuring selection at the level of organismal fitness is a little bit trickier because, as I mentioned, the mutant mtDNA genotype is kind of a moving target. The mutant frequency can vary between individuals and it can vary over time. And so you can't simply compare the fitness of different animals that carry different levels of mutant mtDNA. But you can measure the overall average cost of carrying the mutant mitochondrial genome by competing them against a fixed reference point. In this case, animals that are homoplasmic wild type, so they only carry the wild type mitochondrial genome. And here we see very strong organismal selection, so that the overall takeaway here is that even though different animals carry the mutant mitochondrial genome at different frequencies, collectively they're being outcompeted due to the fitness cost, due to the average fitness cost of carrying the mitochondrial mutation. So, okay, we have this set of experiments where selection can push the mutant mitochondrial genome higher in one context, and it can favor the wild type mitochondrial genome in a different context. And presumably, these different levels of selection keep this mutant mitochondrial genome in a balanced heteroplasmic state. But in order to really know whether or not that hypothesis is true, we need to have a more principled theoretical framework that describes how selection is acting on mutant mitochondrial genomes in order to address the question, do these opposing selection forces actually predict the mutant frequencies that we observe? And so I carried a lot of this data into my postdoc, where I developed a population genetic model to describe selfish mitochondrial genomes. And starting at the intra-organismal level, the mathematical details are here if you're interested, but you don't need to know them to understand the data. Basically, what you're seeing is that this is the intra-organismal fitness. So that's the replication advantage of the mutant over the wild-type mtDNA within any given animal as a function of the mutant frequency within that animal. And so as long as the mutant frequency is low, then the mutant has a replication advantage over the wild type. And then that replication advantage seems to dwindle as the mutant frequency rises. And that's indicative of a pretty common phenomenon, negative frequency dependent selection. It's a pretty common phenomenon whenever there's a selfish or cheater entity in a biological population. And so the black line is the maximum likelihood fit of the model to the empirical data. And then the faint red lines are parametric bootstraps to give us a measure of the uncertainty in the model. At the level of organismal fitness, what we're seeing here is that the mutant mitochondrial genome appears to have a negligible phenotypic effect, where the heteroplasmic animals are basically equivalent to wild type up until you have a high mutant frequency, and then you pass a critical threshold, like I mentioned earlier, where as mutant frequency rises, you get this critical threshold that you pass, and then the fitness of those organisms drops dramatically. And these different levels of selection seem to combine to give us this characteristic skewed distribution that concentrates most individuals toward the higher end of the mutant frequency, which kind of resembles the empirical distribution, at least qualitatively. Okay, so this was all done with a single mitochondrial mutation, right? So we can ask the question, is this how mutant mtDNA behaves in general, or is this specific to this one mutation that I've been working with? And in order to address that question, I'd like to briefly sketch out some possible theoretical scenarios. One is that you have negative frequency dependent selection like I described, but what happens if, as the mutant mitochondrial genome loses its fitness advantage over the wild type at the intra-organismal level, what happens if it loses its advantage at a frequency range in that gray region that is still too low of a frequency to really impact the organismal fitness, right? And what's going to happen here is that the overall distribution of mutant frequencies among individuals that carry this mutation is going to basically be centered on that region where the intra-organismal fitness or selection pressures are balanced. And then the width of that distribution is going to be determined by the strength of genetic drift, which is basically a function of the effective mtDNA bottleneck within any given lineage. I like to call this the flying under the radar strategy, because the selection forces are able to maintain that mutant mitochondrial genome at a frequency that is too low to really have any significant fitness cost on the organism. Alternatively, we could ask, what happens if as the mutant mtDNA loses its selfish replication advantage, it also encounters a strong organismal fitness cost. And here, the theoretical prediction is that we get, again, that characteristic skewed frequency distribution, where, again, the width of that distribution is determined by the strength of drift. And finally, there's another mechanism that I sometimes call constitutive cheating, where the selfish intra-organismal advantage of the mutant mitochondrial genome never goes away completely. Instead, it actually constitutively pushes the mutant to rise in frequency. And as it does so, the mutant mitochondrial genome encounters strong organismal selection. And here we get again this characteristic skew distribution that concentrates most animals toward the higher frequency range. However, the actual shape, interestingly and perhaps counterintuitively, the shape and width of that distribution is relatively insensitive by comparison to the other mechanisms, to the strength of drift. And in other words, the shape of that distribution can be maintained purely by the selection forces that are acting on the mitochondrial genome. So, okay, what about all these other mutations? How do they compare? We have a whole collection of different mitochondrial mutations in different strains of worms that affect different mutations that affect different components of the electron transport chain, different regions of the mitochondrial genome. And to make a long story short, when I repeated the selection experiments and the modeling inference procedure, we found that each of these theoretical mechanisms is represented by at least one of the mitochondrial mutations that we have in our collection. And so, okay, so to round out this first part of the talk, the other parts 2 and 3 will be a bit shorter. We have this general scenario where selfish mitochondrial genomes are able to behave selfishly in the first place because they are exploiting what is normally a cooperative relationship with the nucleus, where the nucleus is able to support, under certain conditions, the replication of the mitochondrial mutation. And this can lead to a sustained heteroplasmic state where the mutant mtDNA is able to be co-transmitted with the wild type for many, many generations and evade organismal selection, either because the mutant is at too low of a level to impose or to subject itself to multi-level selection, or because you have a balance of selection, balancing selection between different levels of scale in the hierarchy of biological organizations. So I call that conflicting level selection, conflicting multi-level selection. I didn't come up with that term. But in any case, the general picture of these different mechanisms is that you have different selection pressures that act in different directions, and that they can maintain these interesting characteristic shapes of the mutant frequency distribution. And the shape of this distribution does in fact contain information about the strength of selection. So the maximum likelihood inference procedure that I used basically fit a model using all of the data that was available. So the measurements of selection at the different levels, plus the overall sample of mutant frequencies. And so the ability to dissect what are the evolutionary mechanisms that maintain these mutations in the population can be better understood in a more precise way by also using information that's available in the shape of these distributions. So, okay, so adaptive evolution, or I would say evolution in general, is of course not just dependent on selection, but also mutation. And this is an area that I've focused on over the course of my postdoc in collaboration mostly with Arlen Stoltzfus and Alejandro Cano. And to kind of illustrate mutation-biased adaptation in sort of an intuitive way, I like to start with some anecdotal examples. So if you mutate A to V at position 63 in the alpha hemoglobin, that actually has been shown to lead to increased oxygen affinity, which has been implicated in adaptation to higher altitude in high-altitude birds like the bar-headed goose shown here. And similarly, if you mutate glutamate to lysine at position 129 of the CYP4 gene in Saccharomyces, that's been shown to contribute to increased caffeine tolerance. And finally, if you mutate valine to isoleucine in position 77 of the HIV protease, that particular mutation has shown up a lot in HIV patients, and as it has the effect of contributing toward drug resistance, specifically resistance against protease inhibitors. And the reason I show all these examples is because the one thing that they all have in common is that they all rely on the G to A transition at the nucleotide level, and or the C to T on the other strand. And this is, in fact, the most common type of single nucleotide mutation, at least in most of the species that we've looked at. And so this represents the general phenomenon that I was talking about of mutation bias, where some kinds of mutations happen with greater probability and at a greater rate than others. And so a big question in the field is, does the strength of mutation bias actually change or affect? Is it a real difference maker in the outcomes of adaptive evolution? And it may not be completely intuitive because, the answer may not be completely obvious because there's a couple of different theoretical scenarios that could be at play. So for example, if the overall supply of new mutations in the population is high enough, then mutation bias might not really matter, because at any given time, you could have a large amount of genetic variation in the population where selection is going to push the fittest mutation to fixation, in which case the strength of mutation bias, again, might not have any effect on the outcomes of adaptive evolution. However, if mutation supply is low, then adaptation works as like kind of a first come, first serve scenario where the first beneficial mutation that comes along has a better shot at going to fixation. And therefore, if mutation bias is very strong, we would expect that to have an effect on adaptive outcomes. So in other words, theory predicts a range of scenarios. And so we need empirical data in order to really settle the question of, to what extent does mutation bias affect adaptive evolution? And so to that end, I've been working on developing this curated data set, which is kind of like basically a meta-analysis by looking at a large number of experimental evolution studies and drug resistance studies in combination with mutation accumulation studies so that we can pair data sets of adaptive outcomes against data sets for the same species of measurements of the mutation rates. And as a first pass, there's a number of different ways we can categorize mutations. But in this example, you're seeing here where mutation bias is represented as the transition-transversion ratio, where transitions are shown in blue, transversions are shown in red. And so that has to do with whether or not those terms are defined, depending on whether or not the purine or pyrimidine nature of the mutated base is preserved by the mutation. And so here we see a very strong effect across the species that are in the data set so far. However, there are a number of different ways that we can categorize and break down the mutation rates and the mutation spectra. And so I did a similar analysis for each of the 6 types of single nucleotide mutations, where beta is basically the shape, the slope of the line, that is to say, on the log transformed data. And so it's basically a measure of the proportionality between bias in adaptive substitutions and bias in mutation rates. And so the null expectation is a flat line where beta is essentially 0. Okay, so we have at least some data that shows that there's a strong signature of mutation bias among the outcomes of adaptive evolution, but what about variation in fitness, right? Adaptive evolution also depends on selection. And so how do we bring selection back into the story? And so to address that question, I'd like to sketch out a quick scenario here where you have a hypothetical mutational target where if we consider all possible mutations that could happen to this sequence, there's going to be some variation in what the mutation rates are and what the fitness effects are. And those can be represented as different values on a number scale represented by mu and s here. And so another way to ask the question is, how do these different factors or how do these different values combine? And to address that question, what I basically did was I plotted selection coefficient or fitness effect against mutation rate. And that gives us, for any defined sequence space where you have a well-defined number of possible mutations that could happen, that gives us a nominal distribution. And then if we allow the process of adaptive evolution to play out, the process of mutation enriches for mutations that have a high mutation rate. So here the circles are resized to reflect the probability of observing any one of those possible mutations. And then selection further enriches for mutations that have a high fitness effect. And what we find here is that the fittest mutation is not necessarily the most common or the most probable outcome of adaptive evolution. And that's consistent with some observations that have been reported in the literature. And the other thing that I think is really interesting here is that this process of mutation and selection can actually induce correlations between fitness effect and mutation rate if you're looking at the fixed outcomes of adaptive evolution, even if there's no intrinsic correlation. And one way to kind of make this intuitive is I like to use the example of food. So I'm a big foodie. If you look at my shopping cart, you'll see a negative correlation between how healthy the food is and how tasty it is. And the simple reason for that is that food that is neither nutritious nor tasty just gets left out of my shopping cart, like candy corn. And so the general gist of the story is that something similar is happening in the case of adaptive evolution, where mutations that become fixed tend to preferentially be some combination of high mutation rate, high fitness effect, or both. Mutations that have neither a high fitness effect nor a high mutation rate tend to be depleted among the outcomes of adaptive evolution. And this is basically just looking at the correlation between mutation rate and fitness effect among the outcomes of adaptive evolution against the underlying nominal space of possible mutations for, I believe this is 1,000 simulated data sets. So the overall tendency is toward the correlation being more negative among fixed outcomes of adaptive evolution compared to the space of possible mutations. we did look at one empirical data set to try to test this idea out. And so to give this one example, single nucleotide mutations tend to occur more often. So if we use multinucleotide versus single nucleotide mutations as sort of a proxy for low versus high categories of mutations that have low versus high mutation rates, then the fitness effect, which is to say the oncogenic effect in the case of cancer, tends to be negatively correlated with mutation rate among patient tumors compared to among the possible mutations in this case in the protein p53. And that's because low fitness effect mutations tend not to be observed. And this tendency helps explain the empirical observation that the most frequently observed oncogenic mutations in actual patients are not necessarily those with the strongest oncogenic effect. Okay, I'd like to round out my time with talking a little bit about taking some of these general concepts that I've mentioned so far and putting them together into these larger, more comprehensive roadmaps of how sequence evolution works using the modeling framework of fitness landscapes. And in the simplest sense, a fitness landscape is a type of map from genotype to phenotype, which can be as simple as the fitness values for two alternative genotypes. And as we explore an increasingly larger space of possible genotypes, we need to accommodate a larger number of possible ways that the sequence can mutate. And so, for example, we can use a square or a diamond to represent the mutational connectivity between four possible genotypes across two loci. And we can use a cube to represent an equivalent landscape that has three loci represented in it. And as we add more possible genotypes, the landscape becomes increasingly higher dimensional. And this general framework, introduced in 1932 by population geneticist Sewell Wright, shows lines that represent mutational paths between different genotypes in sequence space. And to make these models more biologically meaningful, we need a way to infer the overall structure of these landscapes in a way that shows us basically, how we expect evolution to work. And we need a source of the information in the form of fitness measurements. So we need an inference procedure and we need fitness data. And this has been made possible, as I mentioned earlier on, in recent years with the ability to express large sequence libraries in cells, followed by selection on those sequences. And that allows us to obtain fitness values for different individual sequences in really high numbers. And in order to extract biological insights from these data, we need a way to take these raw fitness measurements and infer the properties of the fitness landscape. And in my postdoc lab, we've developed computational methods to do exactly that. And one such method is called Gaussian process regression. This is a machine learning framework, or a machine learning approach rather, that can learn the shape of a complex function based on a sample of observations. So for simplicity, let's suppose X represents variation in genotype and the function represents fitness. We start with a prior distribution of possible functions with no pattern and no data. And as we add some data, as we make some observations of genotypes and fitness values, then the distribution of functions becomes more constrained, and as it becomes updated through a Bayesian process to a more constrained posterior distribution. And this distribution takes on a shape that also allows us to estimate the uncertainty in our predictions. And as we make an increasing number of observations, we can narrow the range of uncertainty in the shape of the function. Okay, with this approach, what does evolution on a fitness landscape actually look like? My current ongoing work addresses that question in the case of an enzyme active site, specifically for the enzyme tryptophan synthase. This enzyme catalyzes the last step in the synthesis of the amino acid tryptophan, and its active site is populated by these four residues at positions 183, 184, 227, and 228. And in a recent study from Francis Arnold's group, they measured fitness on a combinatorially complete sequence library for this active site in an experiment where they selected for the ability to survive in media that did not contain any tryptophan. So it's conditions that select strongly for the ability to synthesize tryptophan. And so this provides the powerful opportunity to test how well do our fitness landscape modeling approaches predict the effects of new mutations that we have not observed. To answer that question, we can infer the landscape using a subset of the data and then predict the fitness of the held out data. So in this case that I'm about to show you here, basically I inferred the structure of the landscape using 90% of the data. And then if we predict the fitness values for the remaining 10% of the data based on just plugging those sequences into the landscape and seeing, what is the predicted fitness based on the structure of that landscape. This is basically what we see. And despite some variation in the data, generally speaking, we get a nice trend where the landscape tends to predict high fitness for sequences that do indeed have high fitness. So we have this model that does a reasonable job predicting genotype fitness, but what does the model actually tell us about how the evolution of the enzyme works? One way to address that is to visualize a low dimensional representation of the fitness landscape. And here, each genotype is a node, one of the dots on the screen, and the faint gray lines corresponds to amino acid changes that can be traversed through mutations. And what this 2D layout does is it captures the expected time that it would take to evolve from one sequence to another. So sequences are spread far apart if, for example, they are on different fitness peaks because it takes some time to cross a fitness valley, or if sequences differ by a large number of individual mutations. And the landscape tells us that there appear to be a few distinct solutions for how to be a good synthase. So for example, if we focus on the sequences that compose this high fitness region here shown in this box, we see an active site tends to prefer the amino acid lysine directly adjacent to glycine. And we can infer similar sorts of epistatic interactions for the other fitness peaks as well. Now, the last thing that I want to mention is that this landscape represents amino acid level effects. But of course, evolution depends on changes that happen at the nucleotide level. And this introduces biases due to the composition of the genetic code. If you're mutating randomly, it's easier to find your way to serine, for example, than tryptophan. There's more serine codons. And then there's also mutational biases at the level of variation and mutation rate between different kinds of mutations. So in my ongoing work, I'm interested in taking these nucleotide level effects into account by modeling protein fitness landscapes in sequence space, or rather taking protein fitness landscapes and rendering them in a nucleotide sequence space. But for now, I'd like to kind of just sketch out a brief glimpse of what that story looks like. And it turns out that the wild type sequence is not actually the fittest genotype predicted by the landscape. And if we map out the evolutionary paths from the wild type to the fittest genotype, we have one of two options, neither of which is very evolutionarily favorable. So one option is to mutate from VIVG to AIVG, which requires crossing a fitness valley by first fixing a deleterious allele before you can then evolve to an even fitter genotype later. And the other option requires two separate nucleotide changes. And one of those nucleotide changes is a T to A transversion, which is among the lowest probability mutations in most of the species that we've looked at, represented by this faint pink band here. And these are the species that we've looked at in my mutation-biased adaptation study that I mentioned earlier. And so it appears that genotypes can become isolated from a more optimal solution, either by selective constraints or mutational unfavorability, or a combination of these factors. And okay, finally, one of my big interests moving forward. So as you know, I have a background in worms, and I'm interested in starting up a worm lab that does fitness landscape modeling, and that has become increasingly possible with the advent of new techniques that I mentioned have been under development in the Phillips group here. And one of those is TARDIS, transgenic arrays resulting in diversity of integrated sequences. And generally how that works is you could think of it as like a high throughput CRISPR, where the sequences that are being introduced into the genome are being sampled from a sequence library. And that allows you to create these really large polymorphic populations of worms that are hypervariable for a specific well-defined edited sequence, which can then be subjected, if you're pointing that approach at functional sequences like regulatory motifs, for example, then you can couple that technique to selection assays that allow you to measure selection in a multicellular animal model, and then map the structure of that fitness landscape to get conclusions about how selection is likely acting on these sequences in the context of multicellular phenotypes. And so on that note, I'm interested in multicellular animal phenotypes. And one of those that I'm really interested in has to do with animal life history traits, which are largely regulated by these signaling pathways that mediate environmental information, environmental cues like nutrient status, to physiological outputs. And this particular example of the FOXO protein encoded by DAF16 in worms is under regulation for its subcellular localization. So it can be either in the cytoplasm or it can be in the nucleus as a transcription factor. And if it's in the nucleus, that tends to have the effect of increasing lifespan at the expense of a reduction in egg laying rate. And these functionalities of FOXO are regulated or mediated through the phosphorylation status of FOXO, which has this, the phosphorylation loci or the phosphorylation sites in FOXO tend to have this consensus motif here. And so I think these represent really good candidate loci for building an integrated understanding that allows us to connect information at a molecular sequence level, at the level of understanding, for example, epistatic interactions between adjacent amino acid positions on one hand, and the evolution of multicellular organism level traits like lifespan and germline development on the other hand. And so on that note, I'm interested in mapping fitness landscapes for regulatory motifs, FOXO phosphorylation sites being one example, but certainly not the only possible example. And then finally, the transition in life history traits, and in particular, reproductive strategy, has occurred multiple times in the overall genus that Caenorhabditis elegans belongs to. And by that, I mean that there's been at least 3 separate independent occurrences of the transition from gonochoristic lifestyle, where the population consists of males and females, towards hermaphroditic reproduction, where the worms are able to self-fertilize. And this has coincided with some really interesting organismal consequences. Like, for example, when worms evolve from being female to hermaphroditic, they don't need males to be able to survive, and so they lose their attraction towards males, which I think is really funny. And so I'm really interested in, I among others are interested in understanding, what are the evolutionary forces that are acting on these genomes. And specifically, another way to ask it in kind of a organismal biology way is, how does the biology of the organism change when it is no longer evolving under the pressure to find a mate? And this is a question of widespread interest, but what I'm interested, kind of the unique angle that I'm interested in is basically trying to map fitness landscapes for important regulatory sequences that play a role in these evolutionary transitions. So to quickly wrap up, selfish mitochondrial genomes, they're very widespread, right? They contribute to human disease, that we find them in worms, and they seem to proliferate because they are able to take advantage of nuclear encoded stress response pathways that are there to regulate mitochondrial replication. And this actually can give rise to a wide diversity of different evolutionary mechanisms that can enable the mutant mtDNA to proliferate. And we see these different mechanisms represented in empirical systems that we can actually study in the lab. Secondly, adaptive evolution, it represents the combined effect of mutation and selection. And so biases in mutation tend to introduce biases in the outcomes of adaptive evolution for reasons that are somewhat independent of the effects of selection. And this can lead to interesting observations, like, for example, the fact that the outcomes of adaptive evolution are not necessarily the fittest, but reflect some combination of mutational and selection constraints. And we see this both in the relatively simple mathematical modeling frameworks, and we also see this in the geometry and the inferred dynamics induced by that geometry of fitness landscapes. And with that, I'd like to go ahead and thank the members of my graduate lab, my postdoc advisor, and all of our collaborators that I haven't mentioned already, and our funding sources, of course. And I thank you so much for the invitation to be here and for your attention. And if there's time left, I'd be happy to take any questions.
Any questions? Sure. What? One.
A really nice talk, and like the combination that you're bringing together in your lab. make a lot of progress. And one of the things I really like is that it begs some fundamental questions, and this goes back to the first section, which is if you've got this this conflict, one way to resolve it is to keep your enemies closer and to move those genes that could actually be part of selfish genetic elements out of mitochondria into the nuclear genome. And so as I was listening, it was sort of thought, I mean, like, why do eukaryotes have mitochondria at all? Why haven't that conflict been settled by just moving everything into the nuclear genome? So a wide-ranging question if you wanted to
Yeah, that's a great oh my gosh, yeah, we could talk for hours about that. That was actually a question that stumped me when I was taking my qualifying exam. Why did these 13 proteins get left behind in the mitochondria? There's a couple of possibilities. One is that these tend to be more hydrophobic among the subunits, and so that could lead to unfavorability, in the biophysics, trying to move them across the cytoplasm. There's some I think it's still kind of controversial, but there's some data that suggests that might be the case. Like, for example, if you take an mtDNA encoded protein and you force it to be expressed by the nucleus, then it can become mistargeted to like the endoplasmic reticulum, for example. Another option is that a little bit closer to your point, that it enables these tend to be the more like catalytically core subunits that are retained by the mitochondrial DNA. And so it has been thought that having those genes expressed in close proximity to the electron transport chain makes it easier to regulate their expression by, for example, perhaps coupling the transcriptional activity of those genes to some readout of the electron transport chain. That's another thought. But yeah, there's a number of reasons. It's pretty controversial, and I don't have a better answer than that. But yeah, sure.
So it seems like your data suggests that there should be heteroplasmy all over the place. And we have some empirical data that reflects on that, but I don't know the answer to that. So what does the natural variation data show?
Yeah, so ironically, I'm more familiar with the natural data in mammals because I think that's where a lot of the biomedical concerns related to heteroplasmy manifest. There are some cases of naturally occurring heteroplasmies in C. brigidae, for example. Sorry, in non-mammals, I should say. In C. brigidae is a good example. There's a deletion, a mitochondrial deletion that occurs in a heteroplasmic state, and it appears to have a replicative advantage. So it kind of behaves like UADF5. And it's been found in geographically isolated wild isolates of C. brigidae. And so that leads to the question of whether or not this heteroplasmic state is able to persist on like a macroevolutionary timescale, or if it's some kind of a mutation bias where it just continues to show up again and again because of, replication slippage. My understanding is that the data seems to favor the latter, but I couldn't tell you for sure.
Is the mitochondrial DNA represented pretty well in Eric Anderson's Sender whole genome sequences?
I haven't, and I need to, because I know that is going to be a really valuable resource, but I'm not yet very familiar with that. I know that there are other instances outside of the context of selfish mitochondria where mtDNA variation in wild isolates becomes very important. There's I believe Joseph Lamelza, who just finished his PhD several years back up in, I think it was University of Washington. They basically did serial backcrossing to take the mtDNA from one wild isolate of the same species and put it in the nuclear background of a different geographically isolated isolate of the same species. And that led to a decline in fitness. And so it looks like there's an incipient speciation event going on that's perhaps mediated by a mitonuclear incompatibility. That's like the kind of the general vein of like what I can say about like what we might be able to find in terms of looking at the natural variation in mtDNA. And there's a number of interesting directions we could go with that, but I still have to take some time to like sit down and really look at what's available in Sender.
Yeah, but even outside worms and beyond, like there's so much
now whole genome sequences being generated and
Yeah.
this is based on our own bias. So and I were just talking about this. Sometimes people don't bother exploring the mitochondria.
Mm.
And so there might just be, many thousands of sequences out there that we could ask what the general prevalence is.
Yeah, when we do generally expect that there's likely to be less variation in hermaphroditic species and less natural variation in hermaphroditic species because we would expect there to be stronger purifying selection on mitochondrial DNA because in the gonochoristic species, if a mutation occurs that's male harming, the mitochondria don't care, right? Because they only rely on the female germline to get passed on. But in hermaphrodites, a male harming mutation is going to harm the whole organism, including the organism that contains the female germline that passes on the mitochondria. So we expect there to be stronger purifying selection. So like, and that's something that to my knowledge hasn't been looked at yet. So I'm very interested in like kind of digging into what kind of natural variation there is in the mitochondrial genome, because that is a very like principled and well-defined hypothesis that we could get the answer to by looking at the data. Yeah. Yeah. Hi.
Here, so
Oh, cool.
totally different perspective.
Uh-huh.
But there's a lot of parallels in your first section between some of the things we think about, right? So if you think about, forces on plant coexistence, and you're competing with your neighbor for water, but then you're collaborating to bring in pollinators. And so we've been thinking about those balances. And it's a lot harder to test in the field, but there's some parallels in the theory, but then we get stumped because there's all this environmental variability. And so we can model it in one environment and then it's different in another. And so then we get distracted building theory around that.
Mm.
So thinking about that, I guess my first question is how much is there, external variability in the rates and processes that you think about? And if there is some, how does the theory develop to incorporate it in your world?
Oh, that's a great question. So when you said external variability in processes, is it?
Is it, or are these, patterns of mutation and the benefits of them totally fixed, or does it vary with cellular conditions? And if it does, how do you guys account for that?
Yeah, there's an incredible amount of variability, right? Yeah. And so for example, one of the experiments that I didn't really talk about here, but I kind of alluded to was that when we introduce like metabolic perturbations, that has an influence on how the mitochondrial mutation affects fitness. And so like, for example, if I wanted to try to translate that into like some kind of a long-term evolution experiment, and I have thoughts on how to do that if you'd like to maybe talk more one-on-one, but like in order to translate that into like a long-term evolution type of experiment, it would be important to introduce something like intermittent fasting where, the phenotype that you're measuring is going to be optimized by selection according to like its ability to flexibly handle different environments, for example. And that's kind of a general workflow that's been done a bit in worms. And I think it provides a really powerful set of like experimental circumstances to try to at least bring some ecological and environmental relevance into the petri dish. Yeah. So.
All right. Well, let's thank Bryan one more time.
Yeah, thank you.
And if you did get snacks on the way in, get them on the way out.